this video, we're going to look at enzymes and how they actually work in the body. Now, enzymes are proteins that speed up chemical reactions in our cells. Now, this is a diagram of a cell. In the center there, we have the nucleus. In the nucleus, we have our DNA, and the DNA actually tells our cell when we need proteins and when to make proteins. And again, remember that all enzymes are proteins. So almost all enzymes are proteins, and they are synthesized and made in the cells. Enzymes help catalyze most reactions that take place in our body. So how many enzymes exist? There's over 3,000 enzymes in the human body alone, and there's plenty of enzymes in other organisms that humans do not have. Plants, fungi, and bacteria also have a wide range of enzymes that humans do not produce, and some that offer benefits for us that we can use to our advantage. So we have bacteria in our stomachs, for example, or in our small intestine, and that actually helps us break down our foods. So they actually help us by producing enzymes that help in our digestion. Or they also decompose organic materials, and that's extremely valuable in every ecosystem. We use enzymes to our advantage, not just inside our body, but here in sewage plants as well. Humans use microorganisms by adding them to sewage treatment plants. And what they actually do is their enzymes facilitate breaking down toxic chemicals such as hydrogen peroxide into oxygen and water. Now, hydrogen peroxide is actually quite toxic, and oxygen and water is not toxic at all. So they're actually cleaning our sewage system. What do enzymes do? This is extremely important to understand. Enzymes bind with substances called substrates at an active site to be broken down into products. Now, in this diagram here, this yellow is the enzyme, and the green substance here, that is called the substrate. As you can see, the green substrate goes and binds with the enzyme, and the enzyme works on it. This section here is called the active site. And after the enzyme has worked on that substrate, it's broken it down into two. So now we have two products. The substrate is now known as products because it's the end of the enzyme reaction. Okay, substrates make products. Enzymes can be used over and over again, so only small quantities of them are required in a cell. How do enzymes catalyze reactions and turn them into products? Enzymes can be extremely specific in their reaction as they have a specific shape with a unique active site. Often enzymes will only bind with one particular substrate, which limits the substrates they can bind to. Other substrates will not actually fit in their active site. So as you can see here, the substrate is complementary to this active site. The substrate enters the active site and binds with the enzyme through weak, non-covalent bonds, thus forming the enzyme-substrate complex. So as you can see, when the substrate binds to the enzyme, it forms a weak, non-covalent bond, and therefore it's able to be released. The substrate will induce a complementary shape at the active site to enable a more complete fit. This strengthens the binding between the enzyme and substrate. I want you to imagine that you're sitting on a beanbag. When you sit on a beanbag, you might wiggle into it so that your bottom actually fits quite nicely. That's how the enzyme and the substrate work together. Okay, this here is a representation of an enzyme. This here is the enzyme molecule, and you can see here an active site. The substrate here, which is yellow, has bound to the active site, and you can see here that there's got a nice fit there. After a reaction is complete, so once the products are completed, the enzyme will dissociate, which means move away from the substrate and changes back to its original shape. Then it can be reused and bind to another substrate. Enzyme specificity. The shape and chemistry of an enzyme will dictate what substrate it will bind with. As you can see here, we've got 3 substrates available, but it only fits a specific type. An enzyme can catalyze a reaction again and again as long as the environmental conditions are favorable. Research has shown enzymes have varying degrees of specificity. So what this means is some enzymes have absolute specificity for one substrate and no others at all, while other enzymes react with substrates with similar functional groups or side groups on a chain. When the substrate binds to the enzyme's active site, weak interactions are formed that cause the enzyme to change shape and fit around the substrate more tightly, forming an enzyme-substrate complex. This middle diagram here shows that enzyme-substrate complex. After the reaction has finished, the enzyme's active site no longer holds the molecules tightly and releases them. So this here is completed, and that's the product. Enzyme structure is very important. When enzymes are actually made in the cell, they're made in primary structure. As you can see, there's a primary structure of just a single long chain. However, it needs to be folded or bent in shape to be functional. So there are different ways that it can be folded. It's called its secondary structure. It might have a helical structure like DNA, like a twisted structure, or it can also be folded, and that's called a pleated sheet. After it's folded in a specific way, they may need to fold further, and that's called its tertiary structure. Multiple proteins make up a quaternary structure, and it's very important for the proteins to be functional that they are actually folded in this specific way. If not, they will not perform their function. So where do enzymes work? Enzymes catalyze reactions both inside the cell, which is called intracellular, and outside the cell, extracellular. Intracellular enzymes speed up metabolic reactions inside the cells only. Extracellular enzymes actually aid in digestion. So when you have the intake of food, it's the extracellular enzymes that help break down the food. Types of reactions of enzymes. What's fascinating about enzymes is they catalyze reactions in both directions. I have mentioned this earlier. So it can go from one large molecule into two smaller molecules, or two small molecules into one large molecule. This here is known as anabolism, and this here is known as catabolism. Naming enzymes. Now, as a general rule, all you have to do is have the suffix "ase" to the actual enzyme for the substrate that it acts on. However, there are exceptions to the rule. These examples that do not fit in this category are pepsin and trypsin, and also amylase. Pepsin is released in the stomach, and it breaks down proteins into peptides, whilst trypsin, that is produced by the pancreas, breaks peptides into amino acids. So enzyme examples, amylase. Now amylase is one enzyme that you produce in your saliva. It actually breaks down starches into more simple sugars. An experiment or a little activity that you can do at home is you get pure bread or even a biscuit which isn't sweet, like a cracker. Put it in your mouth, bread or a cracker, and leave it in your mouth, because the amylase will start to break this down. When it breaks it down, you'll actually notice in a little while the food in your mouth becomes sweet. Your amylase is actually breaking down the starches into simple sugars such as glucose. Lactase. Now lactase breaks down lactose sugar. As you can see, all you do is replace lactose, the OSE, to ase, and it breaks it down into galactose and glucose. Metabolic enzymes are found in every cell in our body. Digestive enzymes. They are named depending on what the group they're acting on. So as you can see here, to our left, we've got fat. Now another word for fat is a lipid. So we've got the enzyme lipase here. Lipase breaks down fats or lipids into glycerol and fatty acids, and proteins are broken down with proteases into amino acids. More examples of enzymes. ATPase is very special. It's another important enzyme used in every living cell. It's especially abundant in our muscle cells. ATPase actually works by removing a phosphate group from ATP in our muscles, thus releasing energy that we're able to use, such as sprinting. You actually have enough ATPase in your muscles to last you for 10 to 12 seconds to exert at your full amount of energy. After 12 seconds of sprinting, you'll find that your body naturally slows down and your body needs to make more ATP. The ATPase works on the ATP to provide you energy from the sugars in your body. You must remember 2 examples for future exams. So here's a table where you can remember 2. I've already mentioned pepsin. Now pepsin's in the stomach and it breaks down proteins into polypeptides, which is smaller proteins. Amylase can be found in the saliva or in the duodenum. Now the duodenum is the first part of the small intestine and it breaks down complex carbs such as starches into simple sugars such as maltose. Now lipase breaks down lipids, which are fats, and it's present in the small intestine. It breaks down fats and oils into fatty acids and glycerol. Next up, we're actually going to be looking at models of enzymes, and it's very important to understand the 2 different models of enzymes. Thank you.